The revitalization and impetus for this project is the release of the Flora of the Southeastern United States 2020 edition by Alan Weakley. For the first time, Alan's Flora that's been in progress for about 15 to 20 years includes all of the state of Florida, which we had been pressuring for several years. As part of that Flora, he also has created derivative or state and region-specific floras for different physiographic regions and individual states. The one for Florida includes only those species that have been known to be found in the state of Florida, with abbreviated keys and descriptions of the habitats and ecology and distribution maps of each species. With this as a baseline, we can then start to think about how we make that flora more specific to the conditions within Florida that these species occur in. Every one of the species in the Flora of the Southeastern United States by Weakley has a little distribution map to go with it, with codes as to whether it's been known to be found in that region, its rarity, and whether it's native or exotic or maybe exotic in that region, and its range outside of the region. This is created from a large database of species records at this level, which enables the Flora Manager software to be able to easily do smaller versions and specific information about each region. Florida has a large flora, and in just the state of Florida, we're dealing with over 4,800 species, probably approaching 5,000 by the time we are finished with the analysis of all of the flora, with the largest families being grasses, asters, sedges, legumes, mints, orchids, etc. Many of these are extremely diverse, and even the genera are diverse within those, such as large numbers of the sedges, carex, cyperus, Rincospora. So the three largest genera in terms of numbers of species in Florida are actually sedges, which means that floristic diversity really is dependent upon understanding how to identify and how to inventory the sedges and grasses that are present within regions. Even in just central peninsula Florida, these are large groups, even in just one region of the state, with on the left panel are the number of species in each family in the Weakley 2020 flora for Central Peninsula Florida, and on the right is the list from the Atlas of Florida Plants on the ISB website from USF. You notice that the Weakley numbers are quite a bit higher. This reflects the use of a modern taxonomic criteria that look carefully at which species should be recognized or tentatively recognized as distinct and not lumped together into synonymy. Those keys can, even though they can be technical, the keys in the Weakley 2020 flora provide a lot of information. If you can see it well enough, you'd notice that the numbered couplets here go from 1, 2, 4, 6, and 9. 1, 2, 3, 4, 6, and 9, missing several. That is because those missing couplets in the key to identifying species lead to species which are not found in Florida. And so the Flora Manager software truncates those keys to only use those characters necessary to identify the species that are present within the state. And again, that group, there's a little distribution map for each species and a description of where it occurs. Those 4 floristic regions that have been used for the Weakley 2020 flora provide a basis for looking at the flora of Florida by regions instead of by a statewide compilation only. The boundaries of these are somewhat arbitrary, and certainly the flora is more continuously variable between these regions. The South Florida region corresponds to that was used by the Institute for Regional Conservation in their floristic survey of Southern Florida. And then the North one and the Panhandle are regions that were included in the previous version of Weakley, 2015. Therefore, Central is what's left between those. Meanwhile, This is a version that I had done of the same family that we're just looking at, the Ericolaceae there, in 1991 to 1994. So I had been sitting on manuscripts of many families towards a flora of the state of Florida that would provide this detailed ecology of each species. I see no reason to continue that as a standalone project when it can be combined with the weekly background information and the structure in order to produce that information using the same structure and the same taxonomy. I've also been doing this a lot and sharing a lot of this online through the Florida Flora and Ecosystematics Facebook group. This is just one example, again, using an area colon as an example of how I posted information about how to identify the species, its history, its habitats, and along with illustrated photos of characters. All of this will end up being part of that Florida flora. And there's plenty to go with. In this case, again, look at the same species and it's like, oh, let's see what thumbnails I have to choose from for photographs. These photographs will be part of the flora, at least selected examples of photographs. What other data sources can we use to determine how rare or how common a species is? I've been doing a lot of work recently in iNaturalist, which is a means of uploading photographs of species from specific locations and having community identifications made of those, and also being able to search for locations and use that information for research. Well, of the species I was just looking at there, iNaturalist has only 4 records for that area colon. And again, you can see where those are located. Surprisingly, let me get back. That's not going to be the entire range of the species, but it's going to give us an indication of how uncommonly it is seen. The Floristic Inventory of Southern Florida by the Institute for Regional Conservation is also a model for much of the information that we're working on. In this case, mostly from about 1999 to 2004, inventories were made of the plants that occurred in each of the conservation areas of Southern Florida, and then their rarity and habitats and distribution were assessed. This is the easily available through the IRC website for all species known to occur in Southern Florida. And it's an example of how we're going to expand this statewide to a larger number of natural areas. And notice they consider this rare in Southern Florida. IRC also developed a ranking system for species at a regional level derived from the Nature Conservancy's ranking system for rare species. If you've seen the NatureServe or NatureServe website, it has G ranks and S ranks for all plant species. G is the global rank. How rare is that species range wide, from a 1 for the most critically endangered species to 5 for secure species. Within states, many species have also been ranked as whether they are critically endangered or imperiled in the state, S1, versus secure in the state, S5. IRC explicitly defined the criteria for these for South Florida as to which species fell in each category and how they determined which category each species was in, and found that many species that were not even on the rare plant radar, no one could relocate anymore within the region. So they were regionally historical or possibly even extirpated from the region. It had been so long since they'd been last seen. And again, another data source for the same plant, we get the county distribution map of specimens from the Atlas of Florida Plants website. And that shows that this is actually that species that's only been seen 4 times from iNaturalist and considered rare in South Florida is known from many counties, including all of the counties of South Florida. So how would we end up ranking this on a regional level? It's G3 from NatureServe. Florida ranks this as S3, also basically rare in the state, rare globally. It is historical only from South Carolina. No one has seen it in South Carolina in over 100 years. And then we'll call it rare, 3 for South Florida, imperiled or a little rarer than that rare in Central Florida and in North Florida, and very rare in the Panhandle. You'd probably in the Panhandle only find this in a few calcareous savannas in the St. Marks to Wakulla area, and very rare in those other counties if you could still find it again. So that's just one example of this project, how we might consider information about each species. We propose to use that weekly subset of the Florida flora to add in detailed habitat information for Florida and differences in habitat between the 4 regions, the rarity or commonness within each of the 4 regions by those expanding their G and S rankings to regional rankings, identification tips about how to tell them apart within Florida, coefficient of conservatism, which I'll get into later, links to the photos of the species, which we can always use more contributions towards, particularly photos that show diagnostic characters of grasses and sedges and other species-rich groups. Eventually, once all this information is gathered and entered, we can work towards a multi-entry key, a visual key, which I will show in some examples of. And then we'll also be working on expanding the keys and identification characters of the species so that we can actually have a better way of identifying and understanding what species we have in a particular area. Some states and regions have done this quite thoroughly. Perhaps one of the best integrated websites of this type is Flora for the Flora of California. And I've shown the data entry screen for that, the entry screen of that on the top left, and a couple examples of the screens you get when you search for a particular species. In this case, I chose Rincospora californica, which is actually rather uncommon, including links all the way to specimens of that species and photographs of it. Another really common and really widely used and very good example of these interactive flora document websites is the Go Botany website from the Native Plant Trust, and which covers the flora of New England. In this case, it has a simple ID method in which you answer questions about the characteristics of the plants you're seeing, starting with very simple ones like, is it a tree or a shrub or a vine or a grass or an herb, and going down through more questions until, and each time it's narrowing down which how many species meet those criteria. And then when you finally get to the species you want, it will give you descriptions, where it occurs, habitats, and photographs. In this case, there's actually even more photographs. I've just got a static version of it there, and its county distribution. Using the weekly flora, there's been an application developed for the flora of Virginia, which actually does that interactive key setup as an app. It doesn't have a web version of that, but just the app version for a mobile device. But the website for the Digital Atlas of Virginia Flora provides also the information about each species, and I've chosen the same one as we were there looking at for New England. Another state that has at least some of this in their state flora information is Michigan. And here, this one from the University of Michigan is not very visual. The key is online, and you can click through the key to plants like in the Cyperaceae panel on the lower left, but you don't get pictures to go with that. And it's really just an interactive version of the key that you might find in a printed flora or a document. But it does, again, go to descriptions of the species, pictures, distribution, etc. This one has another characteristic within it, and I don't know if I can point with my, yeah, I can. Within the information provided called coefficient of conservatism, which I alluded to back in my list of things we're talking about what would be included in the ecological flora, but we didn't go further about. That gets into a concept of how common or rare a species is in fidel or undisturbed habitats as opposed to things which are weedy or generalist. So the more likely a species is to be found in a high quality natural area, the higher its coefficient of conservatism. And the lowest ones would be what we maybe call native weeds or native invasives, species that we don't really consider to be indicative of high quality, even though they might be present in high quality areas. The high quality areas would have many species which were more specific to that. This was really developed in the Chicago area in the 1970s in order to distinguish between different prairie remnants primarily and woodland remnants as to which ones were worth preservation by their floristic composition, indicating that they had less disturbance and more of a species pool for which to recover. And in a fragmented landscape, that became a good way of distinguishing between just an open woodland and a high quality rich area that had lots of native plants. And these are basically ranked on a scale of 0 to 10, and then calculations can be made using those of the quality of the particular landscape at any spatial scale you're looking at, from small plots up to an entire natural area. This has not been applied very much within the Southeast, but there actually are lists of these species and their coefficient of conservatism for South Florida, which I've provided the link to there, and then a partial list for some other regions. Sometimes those partial lists include only a certain set of wetland plants and not the entire flora. But this has actually become fairly widespread in use over a large part of the US and Canada. It's actually been used in Florida a few times. In this case, I'm using coefficient of conservatism and calculating the floristic quality index of different polygons, different community type polygons within the Upper Lakes Basin watershed of Polk and Osceola counties. And you can see, the quality index is somewhat dependent upon the number of species you do have. Areas with more species richness are likely to have higher quality, but you can have high species richness and still have relatively low quality, such as these wetland disturbed and upland disturbed areas, which have high species richness, but their species are mostly weedy, and so they come up with a lower quality index than in contrast to hydric hammocks and mesic hammocks, which had high diversity but many of them were high quality, fidel to natural area species, and areas of scrub, Quercus dominated scrub, and even xeric hammocks, which had few species, but those species were high average quality. You can think of that if you go into a scrub polygon, you don't see but maybe 30 or 40 plant species, vascular plant species, but yet most of them, or a large percent, are narrow endemics or at least regional endemic species, and which you don't find in the general landscape. I've started the process of doing the rankings of these for all species, and I just put up a couple examples to see. And this is the case of the genus Rincospora within Florida, where each one has a different ranking within each region. And in just the process of doing this, I realized some were rarer than we thought. One I came up with right away is Rincospora punctata, which I don't have an in situ photograph of. All I have are specimens. It is G1, meaning globally critically endangered, and state historical. The last time it's been seen in Florida was in 1998, but it still could be somewhere in Northeast Florida, and no one's simply gone to look. So this would be an example of a species we'd try to verify is still present in the flora. I'm going to go back and talk about iNaturalist for a while as a means of collecting information and collecting species records. It's relatively easy to join and set up a profile on iNaturalist. This is my profile as it existed a few days ago, and it indicates what I've put up there. My interaction with iNaturalist is mostly in doing identifications of other people's records rather than my own. I've only put up things which generally have very few or no records on the site, whereas I've done 22,000 and some identifications. I've also established a number of iNaturalist projects. These are collections of the information on iNaturalist for various counties or regions or natural areas. This is a screenshot of that as of a few weeks ago in plants of different groups of counties that I thought would have enough related flora that we could lump together in terms of looking at them, and of some natural areas, in this case, Ecological National Forest and a few others that are getting going. These aren't the only naturalist projects that I have going, and I'm actually members of several others. But in just an example, in the Highlands and Polk County one, there's over 20,000 observations. I'm sorry, this is the Western Panhandle. Over 20,000 observations as of this data side. When you look at those on iNaturalist, you'll see a map of occurrences. The open circles are obscured locations, meaning that the plant has been found somewhere in an area around that point, but they're not revealing the exact location because of the rarity of the species or because the observer wanted to keep that information, sensitive information, obscured. However, it still shows as being present within the area because the boundary of that obscured area is all within the area of the Ecological National Forest. In just the Ecological National Forest, we already have over 5,500 observations by now, and 700 species, which is a very large number for a particular natural area, yet it's still not the total flora of that site. I established many of these projects by uploading or using the boundaries of particular conservation areas, and then iNaturalist assembles the records that fall within that. So this is one for the citrus tract of the Withlacoochee State Forest in Citrus and Hernando counties, which is one we're actively working with. And then another one that was set up by Hannah Katz before I started working with it was one for Lake Wells Ridge State Forest, Arbuckle Tract in Polk County, in which case we've actually curated and identified positive identifications on almost everything that's been recorded from that area. You can search these for location-specific species or for families or for any other taxonomic group. So in this case, I put up the locations of Rincospora megaplumosa that fall within the Arbuckle Tract of Lake Wells Ridge State Forest. So let's talk about some specific species that may be rarer than historical records indicate. Probably one of the most elusive rare plants of Florida is Schwalbe Americana in the Orobanchaceae, the same family as Agellinus and Cimmeria. Being a hemiparasitic plant probably on oaks. It's, you notice the number of county records it has. It's been found in many counties in Northeast Florida and in Central Florida. And the range-wide map indicates also a fairly wide range for the species as a whole. And its NatureServe map indicates that it is S1 or S2 or historical or extirpated in every state that it's known to occur in, that is, has been known to occur in. It went for a while without being seen in Florida, but it's been relocated in the Western Panhandle. So for Florida, this is a G2 species, range-wide. Even with all that number of records, it's almost died out or been extirpated from most of its range. S1 for Florida, Panhandle PFL 1, North Florida extirpated, Central Florida extirpated. I believe the last record for North Florida was in 1940, and the last one in Central was in 1948. We have looked for this plant in some areas where it seems like suitable habitat in Central Florida, and it's just been nowhere to be seen. But that doesn't mean it's not out there somewhere, and it should be on our radar to look for. I'll talk about the genus Platanthra and how those differ in their ranges and habitats within the state. In Peninsular Florida, probably our most common Platanthra, or what people call fringed orchids, but this one's not fringed. I think it's snowy orchid or something like that. I'm not very good with common names. It's got a fairly wide range from New Jersey to Texas, but isn't common in much of that range. This is from the Biota of North America program website, where if it's rare in a state, the counties are colored in yellow. If it's gone from a state or extirpated, it's colored in orange. So this species is extirpated from Delaware and from Arkansas. It's rare in North Carolina, Georgia, Alabama, and it's considered not on the rare plant, and in Tennessee, not on the rare plant list for the other states where it occurs. If we look at the IRC map for South Florida in the lower left, we see that there's a fair number of records in the Martin, Palm Beach County area and in the Big Cypress region. And you can't really expect this to be very comprehensive above the line across that defines South Florida, Charlotte, Glades, and Martin counties. We look at iNaturalist, we find clusters where people observe it. And obviously, it's in more places than this, but it does indicate that we're getting a fairly good number of observations of this plant within the state. So when I end up looking at the rankings of that, IRC ranks it as a 2 for South Florida, so imperiled. But we would consider it at least no worse than rare, no more rare than just a 3 in the other regions of the state, and perhaps even a 4 if we really got to looking at all of the places where it is secure. We'd like to know that it occurs on a fairly large number of conservation lands in order to go from being a 3 to a 4. In increasing order of rarity, we go to Plutantra ciliaris, the orange-fringed orchid. This is much wider ranging, but it's actually a little less ranging within Florida. Notice there's a southern cutoff within the state, and that's consistent with the iNaturalist records, where we have one as far south here. I believe the southernmost I have is Highlands County. This may be actually in Polk, but close to the Highlands County line. But it's not really that common over the entire Central Florida area, and it's a little hard to find south of this line from Crystal River to Daytona Beach, which is about our central to North Florida boundary that we're using. I use an example of it in its natural, in this case in Central Florida, it occurs in cutthroat grass areas often. This is the spatial distribution of one population that's the largest population known of the species, which is in Polk County. So again, this one's a 3 for the Panhandle in North Florida, but 2, a little bit rarer in Central Florida and absent from South Florida. As we move to increasing rarity, we get to Latantra Cristada. Again, we have a fairly wide range. Notice all the way south on the range map to Manatee County. But when we look at the iNaturalist records of this species, we have nothing south of Putnam County. There actually are a few records south of that. And fortunately, by publicizing species like this, we get people out looking for them. And so we have people that have been finding this in counties that don't even have specimens. Levy, Sumter, and Volusia were all new county records of this in the last 5 years by people going out and searching for rare plants as interested citizen scientists. But this is still rare enough in Central Florida that it's a 1 for Central Florida, a 2 for North Florida, and a 3 for the Panhandle. By the way, all of these Platanthra pictures have been taken in Polk and Highlands counties. So we're taking these at the very southern limit of their range, except for Platanthra nivea. Platanthra conspicua is one that most people don't even know still occurs in Central Florida, even though, again, it's got county records as far south as Manatee and Highlands. Manatee, Hillsborough, Polk, Highlands, all it's known from, and several other counties in Central Florida. When we look at the iNaturalist records, there's nothing south of Clay and Baker and Columbia counties. But it still is extant barely in Polk and Highlands, so it's a Central Florida 1. North Florida 2, because it is really relatively restricted to good quality seepage slope habitats. And 3 for the Panhandle, primarily based on how well it's doing in areas like Blackwater River State Forest and Eglin Air Force Base. Then Plutantra Integra. Notice on the wide range map, this one is considered rare in every state where it occurs. It is a G3 species, so rarer globally. S3 because it's actually doing as well in Florida as any other state. But Notice the iNaturalist map. We got shrunk even further down in terms of where it's been recently seen within the state into just the Apalachicola National Forest area and Blackwater River State Forest. And this, which is a general point for an obscured record that probably was obviously somewhere in coastal Bay or Walton counties, not in the Gulf of Mexico. These obscuring of these records by just putting a point somewhere within a radius does enable the records to be hidden from people that might want to go and exploit or dig up this species. But these again are recent photos of this plant from Highlands County. This again is Central Florida 1, North Florida historical, because no one has seen Florida in at least 20 years that I'm aware of, even though there are county records from Nassau, Duval, and maybe one more. Clay. I've had Nassau, Duval, Clay. And but fairly secured in Panhandle, so a Panhandle 3. So I've talked about mostly species which were coming down from the north, but there's also species which come up from the tropics of the southern part of the range into Florida, which become increasingly uncommon to the north. One of these is the pineland heliotrope, Euploca polyphylla, formerly Heliotropum polyphyllum. If we look at its range map, we can see it's very common in the southern peninsula, but increasingly uncommon as we go north. And in the iNaturalist records, that fits that very well. It's obviously through parts of the central part of southern Florida, but people just don't go looking. But it isn't everywhere in this region. As we get further north from that South Florida boundary, the calcareous habitats that this requires become less and less common. So you get it really kind of in some near coastal calcareous areas and not so much inland. We did a little checking of this record here from Dixie County, and it really is it. There's no question this is a valid report, and it is known from the county already. But it turns out I had a specimen record from less than a couple miles from this iNaturalist point already. So this one is one where it's secure in South Florida. South Florida 5 IRC ranks it. Maybe a 3 for Central Florida, 1 or 2 in North, depending on how many more records there are in the Big Bend coast where no one really goes and looks for plants that often in Taylor, Dixie, and Levy counties. And that Escambia County record, is it really real, or is it some introduction that happened and didn't persist? That needs to be investigated as to where that plant occurred, if it was a disturbed area, or was it a natural habitat? We could do this for many sedges and grasses as well. And with sedges and grasses, we find that there aren't as many, we don't get as consistent of records, but we do get some records of these species. One common sedge in parts of its range, but really uncommon in other parts, is Rincospora elegantha. Is really rare in Central Florida and in Northeast Florida, but fairly common in the Panhandle in seepage slopes and seepy wet savannas. That gets me into some other data sources. So in looking for how common that is, I can also look for specimen records. So I've looked for what occurred on Cernak, which is on the left, which is a Southeastern Regional Consortium of Herbaria, which summarizes the herbarium records for species from many different herbaria in the Southeast US. And I get 85 records for Florida, which is a fairly sizable number, even though some of them are duplicates of the same collections. And even 24 records in the Atlas of Florida plants for Florida. In this case, I'm just showing a couple of those, but these are all relatively isolated, relatively specialized habitats. In looking at habitat distribution of species, this is just up as an example. If you have enough records of a species or enough floristic records for a particular area, you can start to see how species vary dependent upon habitat. This is just part of the distribution of the genus Rincospora within the Apalachicola National Forest in terms of some defined habitat types, which I'll go into later on a later slide as to what these mean. But it does indicate that they are not equally distributed throughout these habitats, that some are very specific to certain habitats, others to different habitats. Sometimes there's a species that may be so common in one region that you really wouldn't think of it as rare at all, but it's really kind of uncommon or rare when you get outside that range. The larger leaved wax myrtle, Morella carolinensis, is like that, or evergreen bayberry. It's so common in the panhandle, it's kind of a problem in unburned or thickets along with tie and other species that dominate areas that don't get frequent fire. But it's relatively uncommon in Northeast Florida and is rare in Central Florida. And notice that's actually a disjunction shown because it's really only in a small part of Polk and Highlands counties where the seepage slopes that this species likes are still known to occur. So this one is a 5 for the panhandle, 2 or 3, depending on how many places we could find it at in Northeast Florida, and 1 for Central Florida. This is showing for that southernmost part of the Morella carolinensis range, the yellow triangles show its occurrences on Avon Park Air Force Range, with the crosshatch area being the landscape association of bombing range ridge seepage slopes and the seepage slope landscape of the side slopes of that ridge. And a typical occurrence there where it's occurring within cutthroat grass seepage slopes. So how did I accumulate all of this floristic information? How do we go about accumulating it for larger numbers of areas? This, again, has got more than you can read, but on the left is a list of what I'm considering microhabitat types of Central Florida. And this is expanded and will keep to expand as we look at different types of habitats and what occurs in each one. Then I will create, for a species list from a particular polygon of one of these habitats, I'll create a site code for that, which incorporates the habitat, the location, the county, et cetera. So I'm going to start talking about how we build a site flora from individual polygon surveys using relational databases or using just spreadsheets of occurrences by habitats. So this is just how I build those site codes from information about each location. If you look at a data structure of how this ends up getting computerized, there will be a fairly simple normalized table of the acronym for the species, the site code, presence, and sometimes I like to put the microhabitat code so I can more easily sort by it. Those link to a list of the entire flora and information about each species, to a list of those site codes and information about the date and the community type and other information about that site code and how it was surveyed. And that gets linked to information about the site itself, which incorporates multiple site codes from a single natural area or a single survey polygon. Then sometimes I'll also put out derivatives if I have different information I'm collecting from specific projects, which also relate the site codes to the site code field of this database. I could do a whole talk just on how we work with these relational databases, but at this point I'll just simply say how these are stored behind the scenes. So in looking at surveying a particular area and building a flora from that, I'm going to start with this area in Polk County that basically I'm going to start with this, what's called a calcareous fringing wet prairie and its associated communities. There's this area of relatively calcareous soils between mesic flatwoods and in this case, a basin marsh along the edge of a slough or marsh system. And so as I looked at different microhabitats within that D5 vegetation polygon, I'm getting a number of species recorded from each one, and then a cumulative as I keep adding community types. Then once I've gotten through that D5 polygon, I have 244 species recorded. I'll add the adjacent mesic calcareous / pine savanna, and then the mesic longleaf pine savanna that's not calcareous above it in the B2 polygon. I'm now up to 288 species. Then adjacent wetter savannas, wet longleaf pine savanna, and maybe a little bit into the wetter and almost a wet prairie. I'm adding a few more species. In the depression marsh here, again, that's adjacent to it, I'm adding more species. So that by the time I've finished with this area between this road and the edge of this marsh, we're up to 307 species known. from the accumulation of how many are in each one of these zones. Notice, by the time I get to the maiden cane center of this pond, I only have 16 species in that maiden cane center, but 2 of them are new to the site because they only occur in those deeper depression marsh zones. I'm still in this one polygon, D5 here, and 2 things that were found in the process of doing that. We found 2 species which are quite rare. One is Digitarius Subcalva, which is a G1S1 species, globally critically endangered, state critically endangered, Central Florida critically endangered. Type specimen of this species of rhizomatous, very fuzzy grass is from near Plant City in Hillsborough County, and it's only been seen 3 times since the type specimen, all within this habitat in Polk County. It needs more surveys, but you have to know what you're looking for, and it is mixed in an area with 100 to 200 species, including probably 50 or 60 grasses and sedges. So one really has to scour these in order to find all the little grasses and sedges that are hidden among there. More conspicuously sticking out in the same habitat is Spiranthes igniorchis. Again, this is from that same site, described as a new species in 2017. It's similar to Spiranthes longilabris, but flowering in August instead of in November to December. And since it was within less than a month or 2 after it was described, Chris Evans was able to find it at Kissimmee Prairie Preserve State Park, not too far away, but in a different county. So we were looking over here at this Polk 259M on that previous compilation of flora. And now let's expand out into the landscape further and add in surveys of other community type polygons. As I add in more of these, you notice I'm adding species fairly slowly now, but sometimes in little jumps, but still adding each time you survey more area. As it would be expected, even in an area of relatively similar habitats, as you look at more different microsites and more different survey type polygons, you add more species. The jump from when I went to 205U and 259U here in species is because I'm now into an area. These trees are Nyssa biflora, and we're in a deep basin swamp forest from starting up in our mesic flatwoods and the calcareous wet prairies and the basin marsh. We're now in a deeper basin swamp forest, so we keep adding these microhabitats. As I go a little further out, I add in more microhabitats. So again, we started here, we got to here. Now I'm starting to go up and add the species found in these areas. And immediately I go to a different set of habitats, and I start adding species that weren't found in all of this area together here, because I'm looking at a different set of community types in this case, and actually looking at some intact mesic and hydric hammocks. And so species which are restricted to those mesic and hydric hammocks within this landscape get added to the site flora this allow it. Now I'm up to over 400 species by the time I get through this little block right here. And if I go a little further to this, and now I take this block and then add in this block, we get to 488 species, which mostly corresponds to the area mapped here as this polygon 1B. It's actually just part of that polygon 1B because it's truncated at these road lines right here. But this is relatively similar, which is basically the flora that we have for the Osceola Plain Loamy Circumneutral Flatlands Landscape Association, where we're looking at how the landscape and landscape position and juxtaposition of communities varies within a landscape. And that differs a lot from the typical Osceola Plain, which is acidic flat woods with depression marshes. This has much more connected sloughs and calcareous areas interspersed. So just, again, we got up to 488 species with that survey. If I keep going out and look at landscape associations at a broader scale, I'm not going to go into all of these, but by the time I get to this particular polygon of Avon Park Air Force Range, we're now up to 1,183 species and climbing. Again, we're looking at, in this case, we're looking at maybe now thinking about what occurs in each physiographic region or section of the state. It's a little more complicated to do than counties, but if we have the site locations for all of our observations, they can easily be summarized by both counties and by physiographic regions. And then just examples as physiographic regions. We're continuing to collect new data of this type in order to add to these projects. And this is what it'll take in order to get that kind of level of information for the flora. In this case, my colleague Brett Budak went out on January the 9th and surveyed the flora of 4 polygons within the Royce Ranch unit of Lake Wells Ridge Wildlife Environmental Area, and did a species list, just part is shown there, and a description of the habitats, and then he mapped where each species list was taken from. So each of those 4 colored polygons each had a separate species list done, which can be linked to that polygon then from that point on and used to compile larger floras for that tract of the wildlife management area and for the entire area. Again, I'm going back, that leads me back to the iNaturalist projects, because as they get bigger, they become more useful. So in this case, the Highlands and Polk County one has 27,000 records in it already, and over 200 of those are Dicantheliums, just one genus of grasses, and represent 26 different species within that genus. So we're looking at data sources, and I'll scan through some of these data sources. Obviously, we're going to include the published floristic lists that we have from parks and counties and other areas where people have done a list. And I'm in the process of summarizing those that are available for Florida. As we get those done, we're actually turning them into spreadsheet data that we can actually use to compile from larger areas. In this case, this is a chunk of a master's thesis one by Emily Ferguson in 2004 from Starkey Wilderness Park, and the iNaturalist project to update that we put out that I've created for Starkey Wilderness Park. And then there's also many of these unpublished lists. Most management plans for state parks and state wildlife management areas have a plant species list included. Some state forests do have one also included, and some do not. But these are also being computerized and scanned in order to include in the floristic data for the project. And also, Florida Native Plant Society chapters are doing compilations of field trip species lists and species lists for particular natural areas. The one on the left is a small part of what the Magnolia chapter has available, and the one below is part of one for, I believe that's a Sarracenia chapter for Ocklawahamie River State Park that the chapter is compiling. We know there's many more of these out there, and we want to get them all as part of the project that we're doing for the statewide assessment of the flora. And I have a few databases that are going into this that represent a few 100,000 records over various time periods and spatial extents, but certainly by no means nearly enough to be the floristic composition of the state yet. So I asked for volunteers to start doing flora projects, start compiling the flora of their local areas. And within a few days, I had this list of people here organized by county who are going to try to work on compiling the plants of their particular local natural areas and which areas they're going to concentrate on. It will take having a group of people like this to do this in order to collect all this information. And I believe that all of you can be a part of that as you go out and explore. I have finished my slides finally. And so I'm going to let Valerie decide if she wants to go ahead and entertain questions for me yet, or if we want to look at how some of these websites might work. Okay.
Can you hear me?
Let's get through those. Let's do questions first, just in case, and I may be able to do a demonstration if it's needed to answer a question.
Okay. Sorry, I had to scroll back up to the top. Brandon Corder has a quick comment. He says that if you can now overlay GBIF records, which include various herbarium records, onto iNaturalist maps too. He says it's hidden under the layers, which helps for comparison.
Great. GBIF overlaid onto iNaturalist. Got it. Yes.
Let's see. I mean, this chat is amazing. Kara's in here. Alan Weakley's in here. Alice Mary Herden's in here. Corey Elko, Eric Ungbird, Alex De La Paz, Brandon Corder. I mean, this is the who's who of of botany here. There was
I did not go into the details of how the Flora manager part of this works that UNC and Alan is doing. But that's a whole nother layer of how information is stored, which we're hoping to expand into key characters and traits of species in order to allow people to more easily access multiple entry keys and visual keys.
Kara would like to know if it's possible to get your habitat list.
For what area?
It was back on one of your first slides where you were talking about all of the different like the little subhabitats.
Yeah, someone can contact me and I can send them my Central Florida one. I also have one for the Panhandle that is different than the Central Florida one. And when I've done projects in other states, I also create one specific for those states. Those really should be regionalized, and it's really a matter of trying to look at the landscape in any region you are and see where those breaks occur in vegetation that you can identify and inventory separately. And so the one for Central Florida I've worked on pretty much, and it's good for inland Central Florida.
Okay. Alan Weakley would like to know if your iNaturalist projects are collector projects, or I'm assuming that's in opposition to standard projects. Like
my project, yeah, my iNaturalist projects are all collection projects, and I notice that has limitations now in that it does not include obscured records if their boundary of their obscure bounding box goes outside the area. That's a change that just happened this year. I am a member of some of the traditional projects and a curator of those. And those were mostly set up by FWC. Peter Kleinhans set up quite a lot of these projects that are traditional projects. And Nicole Zampieri has also set up a traditional project for FNA tracked species.
Okay. Kara would also like to know, do you use this relational data used in machine learning for predictive models?
Ooh, that's another next step. Can we develop a predictive model for community composition based on what you see already? Can we tell you which species of Rincospora, let's say, are likely to be at your site? Or which species of Dichynthelium you're going to have within that habitat based upon, a limited species list of shrubs and common grasses. We're not there yet, but that's another goal of the process, is to be able to narrow species identification issues by community composition. I'm not the expert in how to do that.
Callie Adams asks, if we find a plant species that aren't previously vouched for our county, how can someone not formally trained / educated in botany create an herbarium sample?
If it's a place that you have authority to collect the plant, either you're the owner or you know the owner and the owner says to go ahead and do so, you simply take a piece of the plant that has representative amounts of plant parts. So that has let's say for a woody plant, a leafy stem with flowers or fruits, or for an herbaceous plant, maybe a part of the clump where you have the basal leaves, the plant, the flowers, the fruits. You press it, you put it between a newspaper and then press it flat and let it dry relatively quickly if you can. With a little bit of heat, but not too much heat, you get a dried specimen. once you've got that dried, flattened specimen, you can create an herbarium label, which has the information about what it is and where you found it, and include that label with that plant in a newspaper and send or take it to a Florida herbarium, and they will mount it and make it be that county record. It'd be fastest if you're within vicinity of USF and could do it there, but eventually all of them would get entered and digitized. It does not take any special training necessarily to do that, and there's good information about how to do it on the University of Florida Herbarium website from the Florida Museum of Natural History.
Robert Gundy asks, how do we get you plant lists for things that aren't photographed and thus not on iNaturalist?
That's really what we really need most of, because not everything is photogenic, and not everything, you don't want to take a picture of every grass or sedge you see, and or frankly, you don't want to take a picture of Serenoa every time you see it, or every area you see it. So I would suggest making written plant lists that you can tie to locations. So let's say if you go out exploring a particular trail one day, and you linger off and spend time in, let's say, walking through an open pine savanna, just record a list of what you see, put that in either a document or a spreadsheet version, and post and talk about it or send it to me.
And Edwin is pretty accessible, right? You're on Facebook.
I'm on Facebook, and you could you could post some of the information about it if you want to describe what you've seen in particular areas. Yes. Or have questions about some of them. You can also have questions about particular species you've seen and ask those.
And if you are not aware, you can also text me or email me with questions. I check my email during these things. So I have a text question from our policy guy, Eugene Kelly, the one who's going to be presenting, doing the discussion on the Drink and Learn on April 7th. He asks, Edwin, do you envision these data eventually being used to prioritize land conservation projects under programs like Florida Forever?
Yes, definitely. They may end up being more useful at the county or local level, I think, in that the Florida Forever projects tend to think about larger landscapes and not so much this micro level of diversity. But certainly, I think county natural area programs have definitely been using this type of information already for prioritization, where it occurs, where they have the information.
Yeah, and perhaps defending areas that need to be defended.
Or in the case of some areas that may you want to know within a larger area, where are the most special places? Where is the diversity within that area that can't be replicated anywhere else in the area? So that if you did have a management concern within, let's say, a 40,000 acre conservation area, you might know you don't want anything to happen, or you want to pay particular attention to these particular concentrations of high diversity within that aren't replicated.
Dusty would like to know if you would summarize the parts of difficult to identify plants that should be photographed to make it easier if you'd identify them for us.
I'll particularly focus on grasses and sedges, but this applies to a lot of plants. You need a picture of the entire plant, if possible, where you can show its structure. So are all the leaves, that will tell you if all the leaves are at the base or they're climbing up the stem, how big it is, things like that, rather than just a close-up of the flowers. For grasses and sedges, it's often important to have a picture of the leaves at the base of the plant. So fairly close up, so you can see what kind of hairiness or non-hairiness they have. Even bending back a leaf to look at the interior structure of the ligule, which at the junction of the sheath and the blade of a grass or sedge leaf. Generally, I like to see multiple pictures of the leaves in addition to pictures of the inflorescence and individual flowers.
Great answer. And there's a great discussion going on between Alan and Daniel and Robert Gundy of FNA. Robert Gundy says, we at FNA really do use iNaturalist observations when ranking species.
Probably not yet, but I think they will. I think it's a matter of having enough information to do that. Because iNaturalist observations are inherently selective in two ways. They're selective geographically because people aren't going everywhere. People are most likely going to areas that are local to where they are. So you'll always see concentrations of iNaturalist areas around urban areas. And that doesn't mean the plants aren't found in 2 counties away where there's no record of it. The other selectiveness is it's selective by how visible or charismatic the plant is. You have far more records of large and showy species than you do of small and obscure ones. So if you look at what species in Florida have the most iNaturalist records, you get things like Calicarpa, Serenoa, Sabal palmetto, not necessarily the plants that are going to tell you a lot about rarity. But within particular regions, it's really surprising how many records you can get of rare plants. Certainly a lot of people that go out on the Lake Wales Ridge will see the rare plants of the Lake Wales Ridge and photograph them, or anybody going to pitcher plant savannas or pitcher plant bogs would obviously photograph the Sarracenias. Those are all automatically obscured on iNaturalist. All Sarracenia records are obscured for every species and every hybrid. So people can't use those records to find the plants. I went in and changed the settings of every one to make them obscured.
EcoBear 929 asks, I've tried searching local projects on iNaturalist, but I only come up with a very limited selection. Is there a way to search more broadly?
Type in the area that you're interested in, I think. If you've got the name of the area, let's say if it depends on what words people use in the title of the project name. So if you can type a county name, you'll get all the projects that have that county name as part of their project name. That works really well if you have a county that isn't repeated very much in the country, like Volusia, let's say. You're only going to get projects in Volusia County coming up. If instead it's something like clay, you might get projects in 20 or 30 states that have the word clay in them. But that's how I found where the projects previously existed, by searching for the names that they might have in their titles.
Yeah, for local conservation areas, I search for the names of the local conservation areas. And then, not necessarily by county name, because Osceola, you might come up with stuff in the Osceola National Forest.
Yeah, but you'll get a small enough list you could pick which ones you're seeing. Surprisingly, there weren't many for the Osceola National Forest. I had to create one.
Robin says, I'm not finding an iNaturalist project called Flora of the Southeastern United States. What is your specific iNat project where you want our observations?
I'd rather have them go into individual county ones. So let me see, can I?
Yes.
Oh, that's back down here, share screen. Yeah. And then I choose a different screen. Okay, let's choose, where's my browser?
Yes, we got it.
Okay. Unfortunately, they truncated these names, but there are these projects for every group of counties in which they'll collect into those. If you go into a particular, let's say, particular managed area, like in this case, the Ocala National Forest, you could specifically choose to add your observations to the Ocala National Forest one when you choose to add observations. So if I go into edit observations, I don't know how well this is going to work for us, but let's say, let's do something. Okay, there we go. Edit. No, that's not how you do it.
What are you trying to do?
Oh, there we go. This is the screen I want.
Oh, yeah.
Whereas project, on this particular observation, I can choose if I want to add it to a project. I could add this to any of these that are listed as traditional projects. Turns out it doesn't belong. I could add it to Wildflowers of Florida, let's say. And I've just added it to that. I'd already had it in Plants of Highlands and Polk counties. So if this had been from a particular natural area that had a project listed, I could add it to that. If it's automatically added to collection projects. So let's say if I go to the Plants of Highlands and Polk County, these observations are everything that falls within the boundaries that are included. So if I look at that, I get a map that shows me everywhere that somebody has recorded one. It will show up whether they chose to add it or not. If they choose to add coordinates, they can do that. I think I'm moving too fast. There we go. Okay. And so we get those as the ones that simply fall within the county boundaries as part of that project. For a traditional project, you can actually do more, which is to add them directly to the project, like the Carnivorous Plants of Florida one you can do directly to. Okay, this is a traditional project in iNaturalist where there's more stuff that can be done with those. Okay, it's thinking about it. Anyway, I think we can get into a whole thing about how iNaturalist works. It's really good at just incidental observations. It's probably the best for keeping track of larger lists of species. It's more a means I find of looking to see who's going to areas and how often they're going and what they're doing when they're there. What are they seeing? I think it's frozen up on me.
Yeah, it doesn't look very happy. Oh, there we go.
Of course, as soon as I say that, yeah. This is one that actually has more details because it was set up in a way that curators can see more of the observations and work with them and change the identifications. So this was set up by Florida Game Commission, Florida Wildlife Commission. Okay, any other questions?
Yes, Robin is asking, I'm thinking about a student project to monitor Lake Panasoffkee outlet, which is listed as an exemplary mesic hammock in Sumter County. Will our observations automatically annotate to Sumter County?
Yes, your observations will automatically annotate whatever county they fall within, regardless of regardless of what other information you put on it. They'll always be linked to the county's information.
Okay. And we're getting a lot of questions and suggestions to have an iNaturalist how-to lunch and learn and a how to take photos for identification purposes lunch and learn. So
those are both really good ones. I should maybe, do we have time for one more? Let's see. I'm going to go back and let's just go back and do a little search. I'm able to show you an example of how to take. Okay, here's a good example. There's 6 photos here. This was just put up yesterday, if it'll come up, of how to take grass photos for identification that will give you enough information to easily do IDs. I think it's because I'm doing all the streaming and trying to do it at the same time. It's probably not as happy as if I was just doing one thing.
yeah, you're getting a little feedback. Oh, there we go. We're loaded.
So there's a picture of the whole plant. The inflorescence where you can see the spikelets. a closer look at the leaves and notice I can see a characteristic of this plant right off the bat, which is this whitish leaf margin. I know I'm going to verify this ID. I'm happy already. But there's even more where there's a close-up of that very character. Not just Amphicarpum has that. And there's the picture of the ligule. Look at that. The little zone of hairs there. That's more than we can even expect most people to take a photograph of. But if you get down detailed into grass keys, that is a character, and it's done by bending back the blade to expose the junction between the leaf blade and the leaf sheath. It also indicates what kind of soil type it was growing in. And so Dicanthelium tenui is the only thing that has that set of characteristics. I was able to verify that that is Dicanthelium tenui from that picture, that set of pictures. So that's how to really do a good set of grass pictures.
Yeah, thank you. That was really good. Alan Weakley says he needs to clean his fingernails. The plants are more important than clean fingernails. So when you start digging in the dirt, it happens. I agree. Okay, and some day chapter folks are popping in that Joe Monstaoka gave a iNaturalist how-to presentation and that should be available on the Dade YouTube. So I'm gonna hop in and see if it's up. Dade chapter.
Which one?
There it is.
Known as vaccines.
There it is. Okay, I will get you guys the link here in the chat. Okay, and that's all the questions. Anything else you'd like to demonstrate, Edwin?
I don't think I really need to. It's just, if you're on Facebook, come and join the group Florida Flora and Ecosystematics. Here's the information. Got to the top of the page there. You'll see that, which will indicate what you're and probably while we were active here in the last hour, there probably was quite a bit of stuff posted or comments made about it. Over 5,600 members, and it's a high activity group in terms of native plants and native plant habitats. And if you do want to do stuff with iNaturalist, start exploring and just see what you find, and what projects are in your local area or county.
I agree. And you can, start exploring online, and then local chapters are starting to have field trips. And, if you want to be shown these plants in person, get in touch with your local chapter. And
I'm definitely interested in other local chapters' field trip information, particularly if they're going out and collecting species lists while they're on their trip. The Miami-Dade chapter used to do that all the time. And my friend Marty Rossler would record that data and keep a record of it. Even before that, George Avery was doing it. And so they have a large set of information that many parts of the state don't have because of decades of keeping those records.
yeah, if your chapter's not currently making plant lists, I recommend that you encourage them to start on their field trips.
Yes, and you could either get those to Valerie or to Juliet, or you could send them to me as well, and I'll try to I can review them even, and see what maybe you want to look at more closely the next time you go out.
Yep. Alice is kind of giving you crap about pulling the plants out of the ground and getting dirt on your fingernails.
In that case, we're studying those species, and so we actually are collecting voucher specimens in order to do a new revision of the genus Dicanthelium for Florida. So that probably became a specimen.
Yeah, I mean, and you have collecting permits for most of your field sites, so.
In that case, we did have that, yes.
Yeah. So don't go pulling plants out of the ground, people, just so you can identify it. You don't have to.
Yes, you don't necessarily you don't have to. You could get down, but you might not have to get down on your hand. You might have to get down to the ground level and take your pictures very carefully if you want to get some good ones for tiny grasses and sedges.
Yeah, there's a lot of photos of me crouched over on the ground with my camera. And so this is what you need to do to get good plant photos. So get on it, everybody. Well, there's no other questions in the chat, so I'd like to thank you so much for your time and expertise preparing this presentation, Edwin.
Thank you. And for all of people who are still there, I'll be doing a version similar, but with additional illustrations and additional examples and maybe a different emphasis on April the 6th at Lou Gardens for the Tar Flower Chapter monthly meeting at 7 p.m., April 6th.
Great. Yes. I hope I can be there. And if you don't have anything else, Edwin?
Okay, just keep going out there and exploring. And the only thing I'd like that people don't do as much of is keep a record of what you find.
Yes, please. Thank you. Everybody have a great Friday and a fantastic weekend. And thank you for coming to our Lunch and Learn. Please become a member of the Florida Native Plant Society if you aren't already. And if you are a member, we really appreciate your membership. Bye, everybody.